नमस्ते, आदाब, सत श्री अकाल। आज हम बात करने वाले हैं मसल फाइबर कॉन्ट्रैक्शन की। टुडे वी आर गोइंग टू डिस्कस अबाउट मसल फाइबर कॉन्ट्रैक्शन. बिफोर दैट I just would give a brief recap or review of the previous video. We discussed about the myofibrils and the sarcomere fibers, in which we discuss about I band, A band, H zone and so on, the content of these sarcomere. So just few points to be remember. Myofibril are composed of sarcomeres, the smallest functional unit of a muscle. A sarcomere is composed of two different sized filament, that is thick and thin filament, which are responsible for muscle contraction. Myosin, the primary protein of the thick filament, is composed of two protein strands, each folded into globular head at one end. The thin filament is composed of actin, tropomyosin and troponin. One end of each thin filament is attached to a Z disc, and an alpha motor neuron is activated. All of the muscle fiber in its motor unit are stimulated to contract. Now moving forward to our today's topic, that is muscle fiber contraction. We already know that alpha motor neurons is a nerve cell that connects with and innervates many muscle fibers. A single alpha motor neuron and all the muscle fibers, it directly single or collectively termed a motor unit. The synapse or gap between the alpha motor neuron and a muscle fiber is referred to as a neuromuscular junction, that is also named as NMJ. This is where communication between the nerves and muscular system occur. Excitation and contraction coupling. The complex sequence of events that trigger a muscle fiber to contract is termed excitation contraction coupling, because it begins with the excitation of a motor nerve and result in contraction of the muscle fiber, or nerve impulse, or we can say in another word, that is action potential from the brain or spinal cord to an alpha motor neuron. The action potential arrives at the alpha motor neuron's dendrites, specialized receptors on the neuron cell body. From here, the action potential travels down the axon to the axon terminals, which are located very close to the plasma lemma, as we already discussed about the plasma lemma in our previous video. When the action potential arrives at the axon terminals, these nerve endings release a signaling molecule or a neurotransmitter called acetylcholine, which cross the synaptic cleft and bind to receptor on the plasma lemma. This process, referred to as depolarization, and action potential must be generated in the muscle cell before the muscle can act. Now we are going to move forward. What is the role of the calcium in the muscle fiber? In addition to depolarizing the fiber membrane, the action potential travels over the fiber's network of tubules, that is T-tubules, to the anterior of the cell. The arrival of an electrical charge causes the adjacent to release large quantities of stored calcium ion into the sarcoplasm. In the resting state, tropomyosin molecule cover the myosin binding sites on the actin molecule, preventing the binding of the myosin heads. Once calcium ions are released from the SR is the sarcoplasmic reticulum, they bind to the troponin on the actin molecules. Troponin, with its strong affinity for calcium ions, is believed to then initiate the contraction process by moving the tropomyosin molecules off the myosin binding sites on the actin molecule. Because tropomyosin normally cover the myosin binding sites, it blocks the attraction between the myosin cross bridges, and actin molecules. Now, once the tropomyosin has been lifted off the binding site by the troponin and calcium, the myosin heads can attach to the binding site on the actin molecules. Hope it's clear. Moving towards the sliding filament theory, which is the very demanding and I think one of the favorite theory in this all exercise physiology. The sliding filament theory, how muscles create movement. When muscle contracts, muscle fibers shorten. How they shorten? We are going to discuss about all these things. When the myosin cross bridges are activated, they bind with actin, resulting in a conformational change in the cross bridge, which cause the myosin head to tilt and to drag the thin filament toward the center of the sarcomere. This tilting of the head is referred to as the power stroke. आपके गेम्स में होते हैं ना power moves, hidden secrets movements, kind of this. The muscle has a property, or I can say the mechanism, that this tilting of the head is referred to as the power stroke. The pulling of the thin filament past the thick filament shorten the sarcomere and generate force. When the fibers are not contracting, the myosin head remains in contact with the actin molecule, but the molecular bonding at the site is weakened or blocked by tropomyosin. Immediately after the myosin head tilts, it breaks away from the active site, rotates back to its original position, and attaches to a new active site farther along the actin filament. Repeated attachments and power strokes cause the filament to slide past one another, giving rise to the term sliding filament theory. That's all. इतनी सी है sliding filament theory. Now, this process continues until the ends of myosin filament reach the Z disc, or until the calcium is pumped back into the sarcoplasmic reticulum, that is our SR. During this sliding contraction, the thin filament moves toward the center of the sarcomere and protrude into the edge zone, ultimately overlapping. When this occurs, the edge zone is no longer visible. Recall that the sarcomere are joined end to end within a myofibril. Because of this anatomical arrangement, as sarcomere shorten, the myofibril shorten, and muscle fiber within a fascicles shorten. The end of result of many such fibers shortening is an organized muscle contraction. I hope you get the concept of sliding filament theory. If not able to understand, please focus and listen to me and go with your Physiology Simulingam book to get the concept. And if it's still not, please comment on the comment box and let me know where you are stuck, what I can do better for you. I will try my best. Now moving forward to the another topic of the same main title of exercising muscle, that is energy for muscle contraction. Energy for muscle contraction. Muscle contraction is an active process, right, or passive process? Think about it and drop the comment on the comment box. Muscle contraction is an active process. Keep in mind, is it active or passive? Drop in the comment box. I'm waiting for your reply. Meaning that it requires energy. In addition to the binding site for actin, a myosin head contain a binding site for the molecule adenosine triphosphate, that is the ATP molecule. The myosin molecule must bind with ATP, that is adenosine triphosphate, for muscle contraction to occur, because ATP supplies the needed energy. And the enzyme adenosine triphosphate which is located on the myosin head, splits the ATP to yield adenosine diphosphate, inorganic phosphate and energy. The energy released from this breakdown of ATP is used to power the tilting of the myosin head. Thus, ATP is the chemical source of energy for muscle contraction. This process we will discuss in detail in the further upcoming videos to much more in detail and descriptive manner. For now, it is sufficient to know about what is the muscle contraction energy or the energy for muscle contraction. Now, muscle relaxation. Muscle contraction continues as long as calcium is available in the sarcoplasm. At the end of muscle contraction, calcium is pumped back into the sarcoplasmic reticulum, where it is stored until a new action potential arrives at the muscle fiber membrane. Calcium is returned to the sarcoplasmic reticulum by an active calcium pumping system. Again repeating, active calcium pumping system. This is another energy demanding process that also relies on ATPs. Thus, energy is required for both the contraction and relaxation phases. With the calcium is pumped back into the sarcoplasmic reticulum, troponin and tropomyosin return to the resting conformation. This blocks the linking of the myosin cross bridges and actin molecules and stop the use of ATP. As a result, the thick and thin filament return to their original relaxed state. For today, this is our last topic of the video. Furthermore, in detail and with a new topic of these main subtopic, we will discuss in upcoming videos. I'm just giving a refined review what we studied today. The sequence of event that start with a motor nerve impulse and results in muscle contraction is called excitation, contraction, coupling. So we had a discussion about the muscle contraction, what is contraction coupling, right? And muscle contraction is initiated by an alpha motor neuron impulse or action potential. The motor neurons release acetylcholine, which opens up ion gate in the muscle cell membrane, allowing sodium to enter the muscle cell depolarization. If the cell is sufficiently depolarized, an action potential is generated and muscle contraction occurs. The action potential travels along the plasma lemma, then moves through the T-tubule system, causing stored calcium ions to be released from the sarcoplasmic reticulum, that is RSR. Calcium ion bind with troponin. Then troponin moves to the tropomyosin molecules off of the myosin binding sites on the actin molecules, opening these sites to allow the myosin head to bind to them. Further, once a strong binding state is established with actin, the myosin head tilts, pulling the thin filament past the thick filament. The tilting of the myosin head is the power stroke. Energy is required for muscle contraction to occur. The myosin head binds to ATP and ATP is on the head split ATP into ADP and releasing energy to fuel the contraction. The end of muscle contraction is signaled when neural activity ceases at the neural muscular junction. Calcium is actively pumped out of the sarcoplasm and back into the sarcoplasmic reticulum for storage. Tropomyosin moves to cover active sites on actin molecules, leading to relaxation between the myosin heads and the binding sites. Likewise, muscle contraction, muscle relaxation requires energy supplied by ATP.